やります。まあいいんじゃないですか。一応。もうやっちゃいましょう。いいっすよ。いいっすよ。やりましょう。もういきなり僕が始める。あ、あ、そうですか。じゃあやりますか。Okay, we start. So, good afternoon. Welcome to our International Symposium of Life-Nonlife Transition, Boundary Between Life and Nonlife. So this is supported by Japanese governmental grant, Transformative Research Areas B, for 3 years. So primary goal of this Transformative Research Areas grant is to pioneer the new research field through the interdisciplinary collaboration across different scientific disciplines. So as you can see, our primary goal is to dissect the kind of inherent difference between life and the non-life. So this is very, all right. Just a moment. So the super famous picture, Paul Gauguin stated that asked, where do we come from, what we are, and where are we going to? This is kind of long-lasting question of humankind. And to understand what are we, dissecting the inherent difference between life and the non-life will be the key question. So this is generated by Gemini, as you can see. So this is not scientifically correct, though. But this kind of we all know that life and the non-life are different object, and we can make a distinction between those two. But if you asked, okay, then what is the difference between life and the non-life? Maybe you cannot tell too much, very much in detail. Like, of course, the biggest difference is this is living and that is not living. But we human beings have still lacks the definitive answer for this kind of question. So we are trying to dissect this boundary. And of course, this is maybe the humankind have been asking this type of question since prehistoric time. So why now we are trying to ask this question? So we believe that the recent scientific breakthrough probably provides us a new and powerful tool to approach this question. Specifically for us, the recent development of quantitative measurement and analysis of cell death in a single cell level, and recent advances of the artificial cell construction. And maybe some of you know that in 2 or 3 weeks ago, the self-reproducing ribozyme have been reported on Science, and this kind of development of this field is quite rapid. And of course, the revolutionary development of the artificial intelligence, including large language model, helps us to understand what is the difference between living and non-living. For instance, 5 years ago, 10 years ago, maybe no one believed that the artificial intelligence will pass the Turing test, but now, right, all the LLM can pass the question. So with those background, we believe that it will be possible to approach this question in a modern way to dissect the boundary between life and the non-life. So this is schematic illustration of our life-non-life transition. Here's life, and here's non-life, and here's the transition boundary. So you can see this in hyperboloid. This is skewed. So this idea comes from the StatMech, like in a phase transition, there's some universality, right? So even if life and non-life are there's a huge variety, but we believe or expect or hope that if those two transit to each other, there are some universal understanding of that. So we have 4 teams and consist of 6 researchers: the reviving cell team and the cell death team and artificial cells and the theory of self-reproduction. And in a reviving cell team, I am a part of this. Based on our theoretical approach of cell death that I will present right after this opening remark, quantify the living and the death boundary experimentally, and also try to revive the cell doing some chemical manipulation. Then the second cell death team aiming to catalog a variety of dying process and seeking the commonality and the universality of dying process. We'll kill the E. coli and the fission yeast, phylogenetically very different organisms using different stresses like temperature, starvation, osmotic, and blah, blah, blah. And we measure the dying process using omics analysis, spectroscopy, and extract the possible common part of this dying process using dimension reduction or some kind of techniques. Then, of course, this is a little bit naive expectation, though. E. coli and yeast are totally different organisms, but the death process is the very critical phenomena on which living system becomes non-living. So we are trying to see if there's any commonality among that. And of course, those two are top-down approach, killing the cell, and by that, we try to see what is death and what is the difference between life and the non-life. And of course, we have bottom-up approach, which will be done by the artificial cell team. And here, of course, we are trying to construct this artificial cell that is sustainably reproduce itself. And also, we're trying to implement the evolvability on the artificial cell. And also we control their division and also their behaviors. And finally, we have purely theoretical team, which is self-reproduction team. So Richard Feynman said, what we cannot create, we do not understand. That might be true, but the reverse is not necessarily true, right? So if you can create something, it doesn't automatically mean you understand the behind logic. So by studying why it is so difficult, so hard to construct life from scratch using cell automata, including von Neumann's universal constructor and also large language models, we are trying to understand the logic behind. So this is our team, but we are a small group, and we recognize that we still are missing crucial aspects from a variety of biology and the related scientific field. So in this symposium, we invited researchers from a variety of background. So if you see the program, you'll find many keywords like cellular aging, molecular crowding, cognition, and hydrolobiosis, and dormancy and self-organization, open-ended evolution, virus, artificial cell, and also microeconomy, space environment, and the deep marine biosphere. So, of course, the primary goal of this symposium is to learn the aspect of those talks alongside you. So I hope you enjoy and find new thing and exciting thing. So, thank you very much. Then the next speaker is me. So, and you don't have to introduce. So, I'm Yusuke from, well, I didn't say, I didn't do self-introduction. I'm Yusuke from Universal Biology Institute, the University of Tokyo, and also I'm a head of this Life-Nonlife project. So I'm gonna talk about, I wanna measure how much. I'm going to talk about today our approach of theoretical understanding of cell death. So I just told you that our kind of vision. But this is my personal dream to understand the difference between life and non-life. In my research career, I don't know how long it will last, but still I'm assistant professor. To understand the boundary between living and non-living, and also understand why it is so hard to construct life from scratch. And I'm a theoretician, so basically I'm doing that using mathematical approach. So today I'm going to talk about death. So in this decade, thanks to the development of quantitative measurement and analysis of single cell level, there are several beautiful experimental paper on how cell dies and what is happening under cell is kind of suffering to, for instance, starvation or something, and also what kind of phenomenological law can hold for that situation. So, oh, by the way, this is done by Nakaoka. He's gonna talk right after this, not right after me. Then those three papers are from Ulrich Garland's group. He's gonna talk tomorrow, and this paper is from Hyun Yok's group. He's talk tomorrow morning, so don't miss their talk. Yeah, this is just advertisement. So then, but those I like those experiments, but first of all, what is death? How we define death is not very seriously discussed in, of course, experiment, because it's reasonable, because biology is not the science starting from defining something. It's more like observation-driven science. So it's reasonable, but this is not well asked, and also we don't have formal definition of death. So, but we can judge whether your cell, for instance, stressed cell, is living or not. So there are multiple experimental techniques to judge cell viability. So, for instance, the plating assay evaluates the regrow capacity of the cell, and you can measure how much membrane is damaged by doing pH staining assay, and also you can measure the metabolic activity and blah blah blah. But those empirical kind of method to judge cell death can contradict to each other. So here, the authors systematically evaluate the outcome of the cell viability using different techniques. So I skipped the detail, but this is measured by plating, how much they regrow, and this is how much metabolic reaction activity is there. So one of the biggest discrepancy of those two are the result of hydrogen peroxide exposure. So if you expose the cell to hydrogen peroxide, this is very severe stress, right? So if you measure it using the plating analysis, the damage is very severe. So the y-axis is how much damage you get from this baseline. But in terms of the MTT conversion assay, measuring the activity of specific metabolic reaction, hydrogen peroxide basically does nothing. So in a sense, how you measure, which method you use, the outcome or your judgment of living or not living will change. So this kind of motivated me to, okay, this might be a good idea to seriously consider how we should define death, right? Now, then, because I'm a theoretician, and also then what I wanted to say, yes. As a playground, I play around with the kinetic model of E. coli metabolism. Of course, ideally, I want to work on this kind of question using real E. coli, but of course, there are many limitations to experimentally measure all the behavior of the E. coli. So here, then instead of that, I use mathematical model. So this is the model I'm playing around with. This is ordinary differential equation model of E. coli metabolism. So here X is the concentration of metabolites, and V is the reaction rate function, how much reaction happens as a function of concentration of metabolites. And S is the stoichiometric matrix. Basically, it dictates how much metabolites are consumed and produced by proceeding a specific reaction. And in this model, the concentration of metabolites are variable, but the concentration of enzymes are considered to be constant, which is parameter. So, and also this is the, what I want to say, yeah. Cell model, this model, this has the cell growth function. This is complicated, but basically this cell model grows by consuming the metabolites indicated by the yellow arrow. Then a cell grows happily. The metabolic reaction flows like this. Oh, I didn't tell that. This model is the E. coli metabolic model under anaerobic conditions. So it doesn't have pentose phosphate, or oxidative phosphorylation and the related things. So the glucose is taken up from the extracellular environment, and this is metabolized through glycolytic pathway, and some goes to the TCA cycle. This is too bright. Yeah, whatever. Then there are ATP, ADP, NADH, NAD, such. Doesn't help much. 全然ショートはしなくていい。No, it doesn't help. Maybe number 6 will be, yeah. So this is the how metabolism going on under happy condition. And this model has bistability. One state is growing condition that this is exactly the same state that I showed in the previous slide, this one. And this is the growth time course of the growth rate and the metabolite concentration, the log scale. Oops. The growth rate is experimentally reasonable range, and also the concentrations metabolites are kind of somehow crafted in a reasonable range. But if you perturb this metabolic model a little bit, it goes to another steady state that we call the non-growing state. So where the growth rate is basically 0, and concentration of metabolites scatters in a very kind of large range. So then the question is, I want to recall why I play around with this model is that the construct a theory that we can reasonably judge whether this state is dead or not. So the question we have to answer is whether this non-growing state is dead state or just non-growing state. So then specifically, the question would be whether it is a good idea to call a given state dead simply because it has low activity. This is no, right? So maybe you know that the many organisms have dormant state, quiescent state, kind of cryptobiotic state, torpor or whatever you call. They have very low activity, then even under the detection limit. And where the living system are rather very close to non-living material in terms of the activity. It's of course not moving, it's not self-reproducing, the metabolic activity is very low in that state. So we cannot judge the living or non-living simply based on the instantaneous state of the cell, snapshot of the cell. So for which basis should we kind of judge living or non-living? So our proposal of cell death is based on the lesson from plant seed. So I'm working on the unicellular organisms, but for just kind of understandability, I talk about multicellular organisms, but I'm not talking about multicellular. So plant seed looks very kind of quiescent. It doesn't move. It doesn't look very active. But we don't judge the plant seed as dead body. This is probably because we, as an experience, know that if you put plant seed onto the soil and give some water, it starts to grow and bloom, right? So if I say this kind of observation as like scientific manner, we know that if you doing some control, you can bring the seed state to the state that you surely say that, okay, this is living state. So based on this revivability or controllability, whatever I say, we are proposing the mathematical definition of cell death, which is the following. So suppose that we are still dealing with the ordinary differential equation model. So we are in a phase space and X is the given state. So if there's no control from a given state X to the representative living state that I will explain later, by the modulation of allowed control, the state X is dead. So basically, representative living state is state that you are sure to say the state is living. So the reason why I'm saying this kind of complicated definition is that if you are asked, okay, list all the living state it's impossible, right? It's very difficult to judge whether dormant state is dead or not. But with this definition, you are just asked to give me one or two, a few state that you are sure to say this state is living. So if representative living state is given, we can compute the controllability by, for instance, changing the enzyme concentration, gene expression level, or external environment, so glucose concentration, oxygen concentration, and blah, blah, blah. And then if you are able to control back the system to this surely growing state, this state is living. This is our approach. So this state is controllable to this state, then this is living. This state is not controllable to this representative living state, then this is not living. So please note that this boundary is not drawn before computing controllability. First computed controllability, then we draw this boundary. Say that from this state, you are controllable to this state, but you are not controllable to this state. But from this state, you are controllable to this state. Then doing this computation, finally, this region turned out to uncontrollable to the representative living state. Then this is the boundary between living and non-living. So to compute, this is definition, but to compute actually controllability, we have to solve this type of controllability problem. This is called sign-restricted global nonlinear controllability problem. I don't go too much in detail, but this is very difficult class of problem of the control theory. So there's no generic approach to compute the controllability of this type of model. But we newly invented the method to compute controllability, including that we can prove, okay, there's no control. This kind of proof of absence of control is very hard, but we have developed the method. And when I apply this method to this E. coli model, so this is the trajectory going to the non-growing state. And what I'm doing is compute the controllability from each step of this trajectory to the growing state. So I took the metabolite concentration from this point and start controlling the state by changing the enzyme concentration and the external nutrient concentration and blah, blah, blah. This is controllable to growing state. So meaning that it can restart growth. Here, controllable, up to here. After that, it's no longer controllable, meaning that non-growing state is not just non-growing, but also there's no possibility to restart growth. So it's irreversible transition, even though you allow the cell to control the gene expression level. So then, following our definition, this is the dead state. And also, here's the point of no return. And this is just demonstration using a single trajectory. But we can do the same analysis for a number of trajectories. And for each trajectory going to the dead state, there is a point of no return. And we can compute the metabolites concentration at the point of no return. Then we can put the dot, the metabolite concentration state at the point of no return. Then by connecting those point of no return concentration, we can see the hypersurface separating phase space into the living side and the non-living side. That we call this as the separating alive and the non-life zone hypersurface, abbreviated as Sanzu hypersurface. And this Sanzu word comes from the Japanese Buddhism tradition, Sanzu River, which separates the world of life and the afterlife. Right? Okay. So of course, this is conceptually funny, but it helps us to detect which metabolites are important to really measure the Sanzu hypersurface. So what I've done is that I sampled the metabolite concentration slightly below and slightly above the Sanzu hypersurface of that E. coli model. And this is just random sampling. So the shape of the distribution doesn't tell you, I think, anything. But the support, the X region on which the distribution has non-zero probability tells us something. So here the green is the alive side and the red is dead side, meaning that this metabolite, this is acetaldehyde, cannot be higher than the certain threshold if you are dead or something like that. So with the field analysis, we found that those two are consequence rather than cause of cell go through the Sanzo hypersurface. Rather, NADH is important metabolite. So Fumiaki will give a talk right after my talk. Is trying to measure this kind of Sanzo hypersurface based on the microfluidic devices. And of course, I played around with 21 variables metabolic model, so it's not possible to visualize Sanzo hypersurface. But if we use toy model, we can of course visualize Sanzo hypersurface. This gray one is Sanzo hypersurface. So let me summarize. So our proposal is to define cell death purely based on irreversibility, not based on specific molecule, specific something, but if you cannot control the system back to the surely living state, you are dead. Then, and to do actual computation for mathematical model, we have to develop a method, and we have successfully developed a method, and it enables us to compute the Sanzo hypersurface, and we are now measuring it. So, thank you very much.
Thank you, Yusuke. I'm Hidenori Nakaoka, and I'm going to chair this session. And any questions from the floor? Ikegami-sensei.
So, you have a microphone there, and if you want,
Well, thank you very much. It was very interesting. My question is, you said there is a controlling factor function which is operating on this living state, right?
Yeah.
And X equal E operating on this.
The equation is something like this. I skipped all the details, but this is a metabolic model, so there's no gene expression dynamics is included. Also, I'm trying to extend this, but so E is the concentration of enzymes. So this is kind of free controllable. So you can make the concentration of specific enzyme from 0 to 100, for instance. It's allowed.
I see. So my question was, usually when whether controlling living state to dead state or dead state to the living state might be very computationally quite complex and difficult to do it, right?
Yeah.
So that in that case, probably pretty much depending on how you can find a way to bring it back to the living state, right? I mean, usually it's a normal state, normal operation that you cannot do it. So how you can work with this?
So the real computation way?
Yeah, because sometimes it's quite difficult, right? Because people don't know it. That's why they don't understand how to bring it back. But once you are very smart, then there's a way to bring it back, right? So that
with this method, we can overestimate the controllable set to state we are desired to control. So then at least with this method, we can prove the absence of control. And from that, if there's a control, this stoichiometric race tells you how you can control the system back to your desired state. So at least in this metabolic model framework, we can compute the control, how to control. But if we extend to the gene expression dynamics model, still I'm working on it.
I see. Thank you.
Thanks. Brooke?
Thanks. So I really like that you map this formally to a controllability problem.
Thanks.
So the key thing that now keeps revolving in my head is the question of allowable control. And in particular, I would personally argue, and I think it's important that we discuss this as a community, that only environmental control would be allowed. Because if you start sort of saying that you can control internally enzyme levels or so, that's like that's like doing surgery on the cell in a sense, right? And at some point we're going to be able to basically build up a living cell from scratch. It's just a matter of time, right? A key thing I think to consider is that if a cell has no ribosome left, that's an enzyme. Ribosome is an enzyme. Then it's definitely dead because it cannot synthesize a single protein anymore, and it cannot synthesize a ribosome without a ribosome. Right? So it's autocatalytic. So I think, yeah, that's something maybe we should discuss. Like, what
Sure.
is allowable and so
So
on.
the very short answer is that if you allow to control concentration X directly, there's no death because this model is free depicted by the constant X. Then if you control E, then what you cannot control is the direction of the reaction. So enzyme catalyzes both forward and backward. So by increasing enzyme, you increase both together. So this gives you uncontrollability. But of course, I'm trying to extend. I totally recognize that the concentration of enzyme being controllable is too much. So we are now working on it. Yeah, I will. Yeah, we'll discuss later.
Okay, now time